Before we start, I'd like to ask, what do you guys think this is?
A sheep?
A sheep.
Well, this is actually the first mammal ever cloned. It is a sheep, but it's the first cloned sheep, and her name is Dolly. It has a huge scientific outbreak because as humans, we are mammals, and this what showed that potentially humans will also be able to be cloned in the future. Now, my name is Isaac Gutierrez, and I'd like to talk to you guys about the process of cloning. Now, cloning was coined from asexual reproduction from plants and other organisms. It was a huge scientific outbreak in the 80s when Dolly was cloned, and everyone in America will soon be hearing about it more, because as scientists expect to have the first human cloned by 2030, which is coming up very soon.
Excuse me?
Yes?
What is asexual reproduction?
Asexual reproduction, there's many processes of doing it depending on the animal, but it's basically when the animal doesn't have a gender and it'll have a baby by itself, and the baby will have the exact same DNA as the parent. Does that answer your question?
Yeah.
Okay. Now, there are currently two ways that scientists clone animals. This includes embryo twinning and somatic cell nuclear transfer. I will be talking about both of these processes, how they're done, and also what a somatic cell is, so you know how therapeutic cloning is possible. Now, first is the somatic cell. Now, a somatic cell is every other cell in the body besides a sperm or egg cell. Now, the difference between the two is that a somatic cell has 2 chromosomes from each parent, while a reproductive cell only has 1. In order to be used for cloning, it must be a diploid, which means it has 2 chromosomes from each parent. Now, there are about 220 different types of somatic cells, and this means that they contain 2 chromosomes from each parent. This makes it possible for somatic cell nuclear transfer to take place. Without this specific characteristic, it would be impossible because the sperm and egg cells are haploids. Now, next is therapeutic cloning, or also somatic cell and nuclear transfer. In 2014, therapeutic cloning was used for the first time on adult cells. Now, this is huge because it starts off by taking DNA from a somatic cell, they remove it, and then they remove the DNA from a nucleus egg cell, and then they also remove the DNA and nucleus from it. And that makes it possible because the DNA from the somatic cell will be placed into the egg cell, and then it'll soon develop into an embryo. Now, the embryo will then be placed into a mother's uterus where it will grow. The mother will think it is pregnant as normal. She will eat more, be more cautious, but when the baby comes out, it'll just be, and it'll have the exact same DNA as the donor of the somatic cell, and it will also look exactly like the donor. Now, according to the National Human Genome Research Institute, the sole purpose of therapeutic cloning is to create stem cells. Stem cells are cells that can be helped with research to understand new diseases, or they can also help people who have problems that doctors can't help with. This includes if they have any fire burns, and they will place it in the unhealthy tissue to help rebuild it. It can also help with heart disease, stroke, burns, cancer, and even Alzheimer's disease. Now, therapeutic cloning is the more popular form of cloning, but next up is embryo twinning. Embryo twinning is much simpler than therapeutic cloning. The process is much shorter and easier to understand. Well, an embryo is it's an organism before it's alive, basically. And scientists will split an embryo in half, and these will be placed into a mother's uterus. They will cut it at around the breast area, right around here, and each piece will be placed into the mother's uterus. They will each grow into their own unique animal. They will look exactly alike. This is an example of embryo twinning with these 2 puppies. They'll have the exact same DNA, and look exactly the same even as they grow older. Now, in conclusion, cloning is not as complicated as it seems, and we should all know the process to know if we support it or not, as it will be very important in a couple of years when human cloning will be very possible and will most likely be out already.